Hi, welcome to another episode of Tom Kennedy Science. I'm your host, Dr. Tom Kennedy. Now today, this is the first part of a series on cell communication. Before I jump into cell communication, let me ask you something. Do you guys like animals and plants and fungus? Maybe we don't like fungus all the time because we don't really know fungus, and well, fungus does bad things, but it also does cool things as well. But if you're like me, I really like animals. I'm a birder. I go everywhere I can looking for birds. I like reptiles and amphibians. I've been with my friends to the tropics looking for rare lizards and snakes and frogs. And I like mammals too, and I like invertebrates. I just really like animals. And animals are a really interesting life form. We are these multicellular organisms made up of millions, billions, and larger animals like you and I and anything bigger, we're made up of trillions of cells. Now think about this. A cell is the smallest unit of life. It's an independent living thing. But yet you and I and every other animal on this planet are a multicellular organism, right? Made up of billions or even trillions of cells. And to be a multicellular organism, these cells have to communicate with each other. And that is true whether you're a mushroom, a plant. Those cells communicate with each other. And of course, animals. Our cells definitely communicate with each other both nearby and over long distance. And if you don't think they communicate long distance, just watch my hand movements here, right? These are my brain communicating to my hands and to my mouth. They're telling me how to talk and how to gesture with my hands, right? So these cells have to communicate with each other. So in this series, what I'm going to talk about is how cells communicate. Now, what I'm not going to get into is how a neuron functions, and that'll be in a different series. But for now, we're going to do this in three parts. The first part, I want to just talk about today the cell surface of both plants and animals. And I'll throw a little bit of fungus and prokaryotes in there to kind of broaden it a little bit. And then part two, which will be in another video, will be over what's called paracrine signaling, and that's where close by or adjacent cells talk to each other. Then the other one, of course, is endocrine signaling, and that is how do cells communicate long distance. And I'm going to focus there on hormones. All right. Briefly, I always like to take things back, the origins of stuff, that trip back in time. I know, I just can't help myself. But I think about, how did the evolution of multicellular begin? What steps were required to have a multicellular organism? Well, the obvious ones, right? You had to have the evolution of aerobic respiration followed by endosymbiosis. And that, of course, led to the evolution of these larger, more complex eukaryotic cells. The reason why they could be larger and more complex is because cellular respiration is so much more efficient at extracting energy from our environment. So that gave life the energy it needed to be complex. So from there, you got these complex cells, eukaryotes, and they evolved multicellularity. And the way they had to do that, there's a few theories out there about what may have caused that. But at the beginning, they had to learn how to communicate with each other. These cells had to start with some type of simple communication where they could exchange information. And we can actually look to yeast cells, yeast that you bake with, brew beer or wine or mead with. Yep, these yeast cells. Well, they can reproduce both sexually and asexually. So they're reproducing by mitosis, right? Producing asexually, which is two identical offspring. But they also have these mating types where they can reproduce by meiosis and create a haploid cell, which only has half their chromosomes. Okay. But then they have different mating types. They have an A mating type and an alpha mating type. An A mating type will not mate with an A mating type, and an alpha will not, as you guess it, mate with an alpha mating type. So they exchange signals, right? And say, hey, I'm an alpha. Well, I'm an A. All right. Our cells confuse, and we can go from a haploid organism to a diploid organism. So I would imagine that the origin of cell signaling that allowed, multicellular life to evolve started somewhere with some type of this exchange of mating signals. And then as life started living closer together and forming colonies, the amount of signaling, of course, became much more complicated. And then these cells also started responding to these signals in multiple different ways. And one of them, of course, was differentiation. But we're not going to get into that here. What I want to do today is talk about the surface of cells, because it's the surface of the cell, right? Cells are a system. We're an open system. We have materials flowing through our cells, energy flowing through our cells. But a system is all these interconnected parts surrounded by a barrier. And our barrier is our cell membrane. Now, other cells like fungus, plants, and prokaryotes on the outside of that membrane also have a cell wall. But let's take a closer look at the surface of cells. Now, first, you know that every single cell on this planet, I mean every single cell, is surrounded by a cellular membrane. Now, in bacteria and in eukaryotes, that plasma membrane is made up of a phospholipid bilayer. Archaeans, they have a phospholipid bilayer too, but it's actually pretty different from ours. And in some cases, they can even have a monolayer. I know, a monolayer. We will not go into those right now. But we do have this phospholipid bilayer, right? So if you remember, it's got the phosphate heads on the outside, so it's hydrophilic on the outside. Then the hydrophobic tails that are made up of fatty acids turn in toward each other, and you have a hydrophobic interior. And of course, the membrane has what is called selective permeability. What that means is small hydrophobic molecules like oxygen, carbon dioxide, can freely pass through the membrane easily. And so can water. Now, I know you're going, wait, water is hydrophilic. How does it do it? It's small. It doesn't go as fast, but it's small. It can get through it. Now, ions and hydrophilic molecules, they do not easily pass through a cell membrane. Okay? Well, that's important. And ions rarely do. And even large hydrophobic molecules, they cannot really easily get through the cell membrane. So as you learned earlier in the semester, there's a whole bunch of proteins involved with the movement or the transport of materials across the cell membrane, right? You've got the ion channels, symporters, uniporters, antiporters, and you've also got pumps that do active transport. And these help facilitate the diffusion of ions across a membrane or actively pump things across. And these, of course, are membrane proteins. These proteins are what we call a transmembrane protein. And the reason why we call them transmembrane is because they go all the way through the membrane, right? They go from one side to the other. And of course, we're talking about cell communication. So as you can imagine, there are a lot of other types of proteins here called integral membrane proteins. And like I said, these are permanent membrane proteins that are always in the membrane. And like I said, they span it like a transmembrane protein. And a lot of these include receptor proteins, and they include proteins involved with cell adhesion. So they actually connect like the cytoskeleton to the outside of the cell so it can connect it to what we call the extracellular matrix. More on that in a few minutes. But you can imagine that, if you've got a signal coming into the cell, you got to transmit that signal through the membrane. And one way to do that is to have a membrane protein that spans the membrane. So as you can see, and one point that I want you to get here is that cell membranes, these are complex structures. They are doing a lot with these cells, and they are, of course, integrally involved with cell communication. And like I said in the earlier part of the semester, you learned how they were involved with, the movement of ions and maintaining water balance, homeostasis, or even staying out of equilibrium with the environment as well. Now, of course, we know that every single cell is surrounded by this cellular membrane, and it's complicated. I mean, this thing's doing a lot. But there's also what's called the extracellular matrix. So prokaryotes, which include bacteria and archaeans, and fungus and plants, their extracellular matrix includes a cell wall. Now, the cell wall amongst all of these different organisms, guess what? It's different. The cell wall in plants is made up of cellulose. The cell wall in fungus is made up of chitin. Now, these extracellular matrices, they're important, right? Because they give the cell rigidity. They help define its shape, and they also help it to adhere to other structures as well. Now, in animals, we don't have a cell wall. I know, we're like the only one that doesn't have a cell wall. There's a few things out there, and prokaryotes have lost their cell wall too, but all animals have lost their cell wall. But what we have instead is an extracellular matrix. And in animals, our extracellular matrix, I mean, the stuff that actually holds us together, you've probably heard of this protein. It's called collagen. Yeah, the collagen that forms your ligaments and your tendons. Of course, ligaments hold bone together, and your tendons attach your muscle to your bone. Oh, gosh, as we age, it's like we can keep going, but man, those connective tissues start to wear out more quickly than the rest of us, I feel. In addition to ligaments and tendons, collagen is also created outside of our cells, and that's what basically holds us together. Collagen, of course, makes these strong fibers, and it's a protein with a little bit of carbohydrates around it as well. And the carbohydrates make things kind of sticky, right? Now, interestingly, one of the processes of making collagen, one of the final, like, okay, we're going to make collagen and be done with it, requires oxygen. So I always find that interesting that animals evolved after oxygen. And one of the true defining characters of animals, there's a few of them, but one of them is at the cellular level or the extracellular level, we make collagen to hold ourselves together. And of course, we need oxygen to make collagen as one of the final steps. How interesting is that? Now, in eukaryotes, we're making this extracellular matrix. And in animals, let's talk about that for a second. We're creating collagen. Collagen is a protein. You know where I'm going with this, right? Let's go back to our endomembrane system. You recall that proteins are all made in ribosomes, right? Every protein on this planet was made inside of a ribosome. Now, collagen is made outside the cell, or at least it's used outside the cell. That means that collagen has to be made going through the endomembrane system, specifically at ribosomes attached to the rough ER, will synthesize a polypeptide into the endoplasmic reticulum, where it'll get folded and processed, sent to the Golgi body or Golgi apparatus, where it's once again it's going to be processed and then transported to outside the cell, where it will be laid down in an extracellular matrix. Now, depending on the different tissue types, and some tissues and organs have little extracellular matrix, but you have other organs like, let's imagine your bones. Bones have a large amount of extracellular matrix. And of course, we need them because the bones are what's giving us our shape. Now, we use bones for other things besides just giving us shape and for our muscle attachment so we can move. They're also used as a reservoir for minerals as well. So, of course, bone tissue, few cells, lots of ECM, lots of extracellular matrix. And of course, believe it or not, our cells interact with that extracellular matrix. And we do that using integrins, which are these transmembrane proteins. Now, we talked about all these transport proteins, but like I said, transmembrane proteins include these integrins that do other things. One is, we've got an internal cytoskeleton. Remember, it's made up of keratin fibers, actin filaments, and microtubules, right? But what we have to do is, you want to have like skin that's really tough. You've got an extracellular matrix. What you'll do is you'll have this membrane protein that will span through the membrane and attach to the cytoskeleton, right? And then it'll also attach to the extracellular matrix as well. And that's part of what holds us together. Now, also, there's lots of signals going around in a multicellular organism. Lots of signals. They're moving from far away distances, or they're moving very nearby, as in like adjacent cells. Well, a lot of the signaling molecules are hydrophilic, or as the book calls them, lipid insoluble. I don't know why they don't use the word hydrophilic. They mean the same thing, right? Basically, more or less. Lipid insoluble means it won't dissolve in fat. Hydrophilic means it's water loving. Well, that makes sense, right? Because we're sending our signals through our interstitial fluids and through our blood and things like that. So they want to be hydrophilic. But of course, that poses a problem. They won't pass through the membranes very well because hydrophilic molecules don't often pass through membranes very well. So an integrin, or this transmembrane protein, will receive these signals. And, we have lots of different signals, and you have to be dialed into that particular signal, and it will transmit that through the membrane into the cytosol of the cell. So as you can see, these transmembrane proteins have lots and lots of different functions. And one of them is to interact with the ECM, both in anchoring the cell to it and interacting with that, and receiving signals that can transmit it through the cell membrane. Now, plants have a cell membrane. I know we often hear about the cell wall, but they have a cell wall outside of the cell membrane. And that cell wall is made up of a carbohydrate. It's a polymer of glucose called cellulose. Very similar to starch and glycogen, except remember that alpha and beta position of the hydroxyl groups? Yeah, it's amazing how such a small difference in the position of a hydroxyl group can mean the difference between a potato, the starch in a potato, or undigestible cellulose. Wild, isn't it? And of course, cellulose, probably the most abundant organic molecule on the planet. I mean, every single plant cell is surrounded by at least a primary cell wall made up of cellulose. Now, in this primary cell wall that basically gives the shape to a cell, to a plant cell, it's made up of this, molecule called cellulose, but it's also made up with other types of proteins in there, like with glycoproteins. Now, glyco just means like sugar. They form these cellulose microfibrils that form cross-links with these glycoproteins, and it makes it very tough, right? Now, if you've noticed, plant cells can range from like a spinach leaf, which is you can eat all day long and won't hurt you, to as hard as a 2x4, right? And that one, now you'll get a splinter. You can't eat that. So the rigidity of these is based on other proteins as well. Now, before I move on to those other proteins, have you ever neglected your plant and you came home and it's all wilted? That's from a lack of turgor pressure. So a cell wall is also important for giving the cell its shape as well. And what happens is if a cell doesn't have enough water in it, especially water in the vacuole pushing up against it, the plant will wilt. And then you'll water the plant, it'll move water back into the cell, it'll push up against the cell wall, and the plant will become rigid again. So that's another reason why cell walls are also important. But like I said, they can also vary in their rigidity as well. So some plant cells have what's called a secondary cell wall. Now, okay, this is weird. Its primary cell wall is made first, and then you have secondary cell wall, which is made, of course, second. But the secondary cell wall, I know, I often think it was made outside this primary, but it's not. It's made between the primary cell wall and the membrane. It's inside it. And in some plant cells, this secondary cell wall can get really big and really thick. And as I was just talking about, like a spinach leaf or the greens in your salad, with all your lettuce and kale and spinach, whatever else you throw in there, cabbage, you can eat those. You can crunch on those. No big deal. But you can't crunch on a 2x4, right? You can't crunch on wood. And the reason why is that wood has another type of protein in there called lignin. And so if you get these lignified cell walls, that makes them really, really, really strong. And that's why we have trees that can grow, guess what, like over 300 feet high. Even in New Mexico, we have trees well over 100 feet high. Okay, so that's a brief introduction to the surface of cells, right? So part 2 will be on adjacent cell communication, also known as paracrine signaling. And until next time, it's been another episode of Tom Kennedy Science.